I'm Dr. Kristina Deligiannidis, and I'm a professor of psychiatry, OBGYN, and molecular medicine at the Donald and Barbara Zucker School of Medicine at Hofstra Northwell. Our second objective today is to summarize the underlying neurobiology of major depression and postpartum depression, including the importance of the GABA-A pathway and the underlying pathophysiology in certain patients. As you can see from this much simplified figure, there are many biological, pathophysiological mechanisms underlying depression. As depicted here, genetics, epigenetic, and environmental factors influence inflammatory mechanisms, HPA axis function, neuroplasticity, and network functioning. In turn, each of these interact with each other and the neurochemical pathways, including the monoamines, glutamate, and the brain's main inhibitory neurotransmitter GABA. Each of these pathways may contribute to the alterations found in the default mode or salience, affect, and cognitive control networks. This leads to the observed heterogeneous clinical phenotype that we call major depression. The GABA-A receptor is a pentamer. It's formed of multiple subunits, and typically 2 alpha subunits, 2 beta subunits, and 1 more subunit, usually in that 2:2:1 stoichiometry or configuration. And that results in 20 to 30 different GABA-A receptor isoforms or combinations within the central nervous system. And it's really important because the different isoforms determine the anatomical distribution, the subcellular location, and also the kinetics and pharmacology. The synaptic GABA-A receptors are responsible for phasic inhibition, and the extrasynaptic GABA-A receptors, usually having a delta or a specific alpha component, is more responsible for tonic and spillover inhibition. Now, neuroactive steroids are natural or synthetic steroids which interact with membrane-bound neurotransmitter receptors. The individual GABA-A receptor isoforms, again, are important as neuroactive steroids are somewhat selective in the GABA-A receptor they bind. Mainly, they bind to the alpha subunit on the delta containing receptors, as depicted here. Many neuroactive steroids are positive allosteric modulators of the GABA-A receptor, again, enhancing tonic or phasic inhibition by facilitating negatively charged chlorine ion flow. GABA that's released from vesicles rapidly activates a family of postsynaptic GABA-A receptors by binding the GABA binding site, also seen in this figure. And that gives rise to a phasic response. And then the neurosteroids that can be released locally from neurons or glia help to prolong these responses, enhancing synaptic inhibition. In comparison to other GABAergic agents, such as benzodiazepines do not modulate extrasynaptic delta-containing receptors. Here we see neuroactive steroids and their relationship to GABA and glutamate and depression. The stress neurocircuit represents a subcortical cortical neurosystem within the HPA axis that regulates arousal, neuroendocrine, and affective responses. Neuroactive steroids have important roles in the HPA response in both acute and chronic stress conditions, which you'll see here. In euthymic mood states, here on the left, GABAergic inhibition and glutamatergic excitation are balanced. Acute stress results in imbalances between GABAergic and glutamatergic signaling. Stress-induced increases in glutamate may lead to abnormal GABAergic transmission. In addition, stress-induced abnormalities in GABAergic inhibition can lead to chronic HPA axis activation. And then with chronic stress, that can lead to a gradual downregulation of glutamate receptors. Concurrent impaired GABAergic inhibition prevents the recovery of those glutamatergic synapses once the stress has resolved. And so during depression, GABAergic and glutamatergic transmission may be balanced, but the signaling is at lower levels. The evidence for HPA dysfunction in both MDD and PPD suggests that normalizing HPA axis function may be a therapeutic target approach for these disorders. In animal models, allopregnanolone and analogs of allopregnanolone rebalance neural connectivity rapidly and in a sustained way. There's evidence that allopregnanolone acts on those delta subunit containing GABA A receptors to shift the network to a healthy network state that is more stable and can persist in the absence of the compound.